Um, Snappy entrance. Hi, everybody. Today we're going to be looking at the structure of a gene. And I really want to talk about this because I think people's understanding of structures of genes don't really jive with what really is there, and I think it causes a lot of problems in understanding. So, if you look up here at my little diagram, what you'll see here is two different genes. This one up here is a prokaryotic gene. And what you can see is that this is a pretty 2D structure. It looks like almost like a line or a tube or a column from left to right here. You have different pieces of the DNA sequence highlighted. And so you can see I've got blue here, light green, green, and so this is representing a prokaryote, a gene that you would find in a prokaryote, like a bacteria. And what it's saying is that there are regulatory sequences here in light green. There's a coding region here, and this would be the part that codes for the amino acids, and then a termination transcription region here. So essentially what would happen is this would be fed in, or an RNA polymerase would come and bind somewhere here. It would read this gene from this way, produce an RNA transcript, and then detach here. That's how prokaryotic transcription works. That RNA transcript would then go to a ribosome where it would be translated into a protein. Now, eukaryotes are different. Here you see, again, we have this like two-column structure, and you see a much bigger regulatory region here. We have our coding region here, but you can see it's divided. Like this is a coding region, this is a coding region, this is a coding region, and this is a coding region. And what that means is that these little bits in the middle get cut out. So these bits here are not coding regions. That doesn't mean that they're useless. They do lots of other things, but they are certainly not going to go and contribute to the structure of the amino acid chain that this gene is building. So what would be different here is that there would be a bunch of proteins here in the transcription area, the regulatory region, or what we call the promoter, which would come and bind. They would be RNA polymerase would be one of them, but there would also be transcription factors and things called enhancers, and we'll talk about enhancers and activators here in just a minute. So this whole structure, a bunch of structures, would come together to form a transcription complex, and then that transcription complex would be activated by what looks kind of like a scorpion coming to sting it. That would shoot RNA polymerase off down this. It would transcribe, RNA polymerase would transcribe this whole region until it got to the terminator, and then that mRNA sequence would be spliced by something called a spliceosome, and the intronic regions would come out. The exonic regions would travel outside of the nucleus and then go to a ribosome where they would be made into amino acids. So we're going to take a closer look at some of those things. So first of all, let's look at this structure thing. So here we're looking at the DNA here. We are looking at what would be the promoter here, and you can see some of the early proteins coming and binding to the promoter. One of these, I think the big green one right up there in the front, is going to be RNA polymerase. And it's this movement here that I kind of want you to take a look at, where we have the back end of the DNA kind of folding up, and you see the scorpion tail-like structure that's formed here. So what we're looking at is this kind of structure here. This is your DNA. This is your RNA polymerase. These are several of the proteins that you saw, and these are what are called distal control elements. So these are pieces of DNA that are a piece of DNA that is far upstream, like thousands and thousands of bases away from this promoter, and they're going to still help to determine when this gene A is made. So activators, and you can have proteins that both activate and work against transcription here, will bind both to this transcription complex and to these distal control elements, causing this little like loop bendy thingy. And once this loop bendy thingy happens, the RNA polymerase will start to transcribe. What's cool is that each of these pieces can come or be activated by different processes. So sorry, I got interrupted there. So basically the point is that I could have, for example, 6 different energy sensors that activate the outside of a cell, activate receptors on the outside of the cell, that were all necessary for gene A to be made. And let's say gene A is going to be helping us to store energy in the cell, which you wouldn't want to do, which a cell would not benefit from unless there was quite a bit of energy around. So you end up with these if and then situations. If these 6 things are present, then you get transcription of gene A. And these things are only going to be present if there are several upstream events that go ahead and activate that. And then gene A is going to do something, which means you essentially have a situation where you have like these 6 things must occur, and then energy storage will happen. Let's take a look a little bit closer at what that looks like. Okay, so here we have the activation of transcription. There goes our RNA polymerase. You can see it is traveling down a piece of DNA here. It is spitting out an RNA segment. There it is. It is spinning really, really fast. DNA is being fed in here. It's coming out here, and nucleotides are around here in yellow. This is the RNA transcript, which will then be, in our scenario, be spliced. Let's see if it shows that. Probably about here. All right. So there's our RNA transcript. Nope, didn't get spliced. Oh, there it is. It did get spliced somewhere. Usually when you see these little loops forming, that's about when it's about to get spliced. Those loops are getting it cut out. Here it looks like they've cut that piece of the video out, but you can see the RNA segment leaving the nucleus here. It's going to travel to a ribosome, and that ribosome here you can see is going to assemble on the RNA, and it's going to start to build amino acid chains called proteins, and those proteins are going to go ahead and do whatever we want them to do. Some examples of proteins are, for example, tubulin, DNA polymerase, I don't know, proteinase. That's actually protein. Anyway, so that's going to be parts of a gene for us. The super kind of important thing is that if you look at a real gene, so let's go to, this is actin, which is a protein, and we're going to view this in the UCSC gene browser. So I'm going to just click on one of these reference sequences and pull up the gene itself. So what I want you to look at is that we are going to look specifically along these with these what do you call it, browsing parameters, is that right? And that's going to show us some pretty interesting stuff. Okay, so we're going to go here, we're going to go exons in uppercase, everything else in lowercase, and you can change that if you want to, but here we go. All right, so you see we have exons in uppercase, everything else in lowercase. Wait a second. This is a bad example. So what we have here is a RNA polymerase. This is going to be on chromosome 5. What I want you to look at here is that we're basically putting exons in uppercase and everything else in lowercase, which means the coding region, the parts that are actually going to get coded into amino acids, are going to be the only thing capitalized. And so what I want you to see right away is that as we scroll through these things are absolutely massive. And what you end up with is these little segments within these huge genes that end up being exons. So I'm just scrolling here, and these are thousands and thousands of bases long. here we go. We found another exonic region. It looks like we go down to the end here. But what I want to show you with this is that these intronic regions, the regions that we're seeing here, this intronic region, they're huge. These promoter regions are huge, and these exonic regions are really, really much smaller in comparison. That means that if a mutation happens somewhere, it's most likely not going to happen on this coding region. Anyway, that's I think what I need you to know from basics of a gene. Good luck.